All right, time for chapter 4. So chapter 4 deals with genetic stuff, so doubling of our DNA, making a copy of it, making proteins, and also going through the cell cycle. So looking at DNA, which stands for deoxyribonucleic acid, so there's your DNA, has all of the genetic material inside your body that your body needs. So even in different cells, it'll have all kinds of instructions for every cell, and only certain genes will be used for that cell. So it's not all used in every cell, but every cell has all of the DNA blueprints for the body. So inside your nucleus, there are 46 chromosomes, which are your DNA molecules. You get 23 of them from each parent. Your DNA, as well as your other nucleotides, are polymers of nucleotides. So the nucleotides are our monomer building blocks to create our genetic material. So each nucleotide consists of a sugar, a phosphate group, and a nitrogenous base. So the sugar and phosphate group is what's common across nucleotides for either DNA or RNA, and then the nitrogenous base is what differs between the four of them, excuse me. Okay, so here's the structure of it. We have phosphate group on this side. Here in the blue is our sugar, so we're looking at a DNA nucleotide because it's deoxyribose sugar. In our RNA molecules, the sugar is a different one. It's called ribose sugar. So that's where we get the R for RNA. And then here's the part that makes our nucleotides different, which is our nitrogenous base. So for DNA, there are 4 bases. So these are our 4 different nucleotides that will make up our genetic code. So depending on its pattern will tell us which amino acids to use in order to make our different proteins. So we have 2 different types. We have our purines and our pyrimidines. Our purines, which are adenine and guanine, have 2 carbon rings on them, so these 2 up top. And then our pyrimidines only have a single carbon ring on them, so that's cytosine and thymine. So these 4 up top are the ones that are part of DNA. This last pyrimidine here at the bottom, which is uracil, that one is only found in RNA. So it actually replaces your thymine in RNA. So the structure of DNA is in a double helix shape, so it looks like a ladder, and then that ladder is twisted like a spiral. The side piece of the ladder are your phosphate groups and your sugar, so phosphate group and sugar. And then in between, which is what we call our rungs of the ladder, are our nitrogenous bases. So each of these steps or rungs in the ladder are paired bases, so they have a particular partner that they always pair with. So in order to unite the nitrogenous bases, they do so through hydrogen bonds, so it's a weak bond that's easily broken. So when you need to open up DNA, you can easily open up your DNA, read your genetic code to make your RNA copy of your gene. So on your strands, a purine is, on one strand, is always bound to a pyrimidine on the other side. So in the case of DNA, the adenine will always pair with a thymine. So this purine pairing with this pyrimidine. And your guanine will always pair with a cytosine. So these actually have 3 different hydrogen bonds between that ones. So when it comes to pairing, let's say when you're making a copy of DNA and you accidentally put the wrong nucleotide with the wrong with a different original base pair, because of this pairing, it actually causes the shape of the DNA to be wrong. So when you're having to go back and do the corrections, you're able to determine that the base pairing is incorrect because it actually makes a kink in the DNA because the matching is off. So again, for our DNA base pairing, adenine and thymine, or A and T, pair together. Cytosine and guanine also pair together, so our C and our G. So if you had a strand of DNA and you only knew one half of the strand, you'd be able to still code for the other. So in this case, if we're looking at what the code should be on this other side, so we know, okay, a thymine should pair with this one, a cytosine should pair with this one, a adenine should pair with this one, and another cytosine should be here. So our code for our matching strand should be T, C, A, C, which is this, T, C, A, C. So they're going to match always. So this is called the law of complementary base pairing, where the sequence on one strand will determine the sequence on the other strand. So when you put these sequences together, some parts of your DNA actually code for a protein or multiple proteins. So this segment that actually codes for proteins is called a gene. In the human genome, which is all of the genes in our entire DNA, we have about 20,000 genes. Those genes only account for about 2% of the total amount of DNA. The rest is what's called non-coding DNA. So it's either to help our chromosomes be in the right shape and structure when they're compacted into the kind of X-shaped paired chromosomes, or it's help with regulating our gene activity. So there's certain pairings either before the gene or in the middle of the gene or on the end of the gene that allow us to turn the gene on or off depending on what cell that DNA is in. And so you can have spaces where you might have something that binds to it to prevent that DNA gene to be copied into RNA. And when you remove that blocker off of it, then you're able to transcribe that, transcribe that gene into RNA to be able to make your protein. So with the massive amount of DNA that is in our nucleus, in order to have some order and structure on our DNA, so it's just not a bunch of strands just loosely pushed into our nucleus region, there is a way to compact DNA so that it is organized. So we'll start with our least compact, no kind of organization, just the basic strand. Take a whole DNA strand, it's about 6 feet long. So in order to start packing this in and make our very long molecule into something that can fit inside the nucleus of a cell, first type of compaction is creating what's called a nucleosome. So it's DNA wrapped around a histone. So here in this little blue circle, that's a histone protein. So when you wrap the DNA around it, so it's roughly like 1.5 times to make to go around that histone, and then you have a little bit of linker DNA to the next section. This whole section is called a nucleosome. So it's the histone and the DNA that's wrapped around that histone. These histones are then folded in a kind of like accordion shape to zigzag, and helps to compact the strand even more so. Then the last level of compaction, which is normal just in a regular functioning cell, is called chromatin. So you take these little zigzags and then we fold them up again into what's chromatin. So in this level of compaction, the DNA is still readily available to be transcribed into RNA. So you're able to go in there, find that gene, open it up so you can make your copy, and then put everything back together. If your cell is going through a division, so creating a new cell, then the DNA is packed up even further so that as you are moving the DNA into your two different cells, so you're going to make a copy of that DNA, and then you're going to pack all that DNA away so that you'll have DNA for both of your new cells. The chromatin is packed in what's called a chromatid. So here in this little X shape, one side is one copy of that particular section of DNA where it's been wrapped up, kind of like an old school telephone cord. So it's highly packed up into this chromatid. So just one side, that's one copy of it. So for example, like from your mom. And then the other side is the other copy from your other parent. So one parent's copy and then the other parent's copy. So this is bound together to each other through a structure called the centromere. So this whole thing is still called a chromosome. So it's a double-stranded chromosome. You also can have single-stranded chromosomes as well. So when the cell is going to divide, again, the cell makes a copy of all the nuclear DNA. So the DNA that is located inside the mitochondria is not copied. You do make more mitochondria so that you can have enough organelles for both new cells. But for looking just at the DNA that's in the nucleus, that's only what's copied during the S phase of the cell cycle. So during this phase, after you've copied your DNA, you've packed it into your chromatids. So again, one copy from one parent, the other copy from so one copy from one parent, one copy from the other parent. So these are identical copies of the same set or chromosome of DNA. So when they are packed up together like this, that's when you're able to see them with a light microscope. So one of the things we'll do in lab is we'll actually look at some cells that are going through division and seeing the cells that actually have their chromosomes compacted together, and we'll be able to see them with our slides. We'll also look at the different phases as the cells, different cells are in different stages of mitosis, and able to see the changes that happen as you're taking your two sets of DNA and moving your DNA to your different cells. So your other type of nucleic acid inside the cell is called RNA. So your RNA is different from the DNA only through two different ways. So it's through, instead of deoxyribose sugar, it's ribose sugar for RNA, and your uracil replaces your thymine as your base. So instead of that A being paired with T, in this case, A is now paired with U. Your RNA is also significantly shorter than your DNA, usually because your RNA is only a copy of a particular gene, so that's usually a whole lot shorter than the entire DNA being copied. So there's three different RNAs that can be made, and they all have a job in making proteins. So the first one is our messenger RNA. Our messenger RNA is the copy of the gene. The second type of RNA is what's called the ribosomal RNA. So it reads the messenger RNA to look at the pattern of the nucleotides to code for your different amino acids. It also provides kind of like a scaffold for making your protein chain. Last type of DNA is called your transfer RNA. Last type of RNA is your transfer RNA. Your transfer RNA is the one that will bring the amino acids to the growing peptide chain that's on the ribosomal RNA. So it's providing that scaffold. The transfer RNA is going to bring those amino acids to that location and pair up the correct amino acid with the particular code on the messenger RNA. So with your RNAs, they also are one nucleotide chain, so only one side of that ladder, so it's not a double helix. And it functions mainly in the cytoplasm. So ribosomal RNA and your transfer RNA or tRNA are only located, are primarily located in your cytoplasm, ribosomal RNA being made in the nucleolus and then transported out of the nucleus. Then your messenger RNA is made in the nucleus, So opening up the DNA, copying your gene, and then that's transported out into the cytoplasm. So here's just a chart to compare your DNA versus RNA, looking at the differences in the sugar, your base pairing. So here again, our U is replacing the T number of bases, so a lot shorter in RNA. 2 nucleotide chains, 1 nucleotide chains. DNA stays in the nucleus, RNA leaves the nucleus, and your DNA again holds all of your blueprints for your proteins in the body, and then your RNA creates those proteins. So a gene is a segment of DNA that carries the code for a particular protein. Our body can create millions of proteins, but we only have 20,000 genes. So the reason for this is that we have genes that can code for multiple proteins. So they'll either synthesize one or more proteins. So the sequence of our amino acids to build the protein is determined by the nucleotide sequence on the DNA. So here at the bottom, we have our gene that's going to code for our protein or proteins, depending on what we're going to make. So the gene is eventually going through the process that's transcribed into the RNA, and then that RNA is translated into our different protein products. So you can make multiple proteins from the same gene. So it just depends on what happens during the process of making that protein on which protein you're going to make. So you could make the entire thing and then cut it and put stuff back together to make a different protein product. So the genetic code on our DNA, again, can make millions of different proteins, and that is called the proteome. So our genome is our genes, our proteome is our proteins. So we make our proteins from only 20 different amino acid building blocks, and this is based off the code from our 4 different nucleotides. So the genetic code is the system that codes for our 20 different amino acids to make the pattern of where our amino acids are going to be placed into our protein. So the minimum amount of code to symbolize an amino acid is 3 nucleotides. This is called a codon. So here's our 3 nucleotides here, and it's going to code for this particular amino acid. So you can make a whole lot more combinations of your codon codes. So some of your codons are going to code for different amino acids. So again, your base triplet, so your 3 DNA nucleotides will code for 1 amino acid. That 3 base sequence in the mRNA, again, is the codon. There's 64 possible codons to represent 20 amino acids. Some of them are for an amino acid themselves. There's 3 that are stop codons, so that's your end of signal, which are these 3. And every gene starts off with what's called your start codon, which is AUG, and that is your first amino acid, regardless of whatever protein you're going to make. That AUG codes for the methionine amino acid. Depending on the processing of your protein, that part can be cut off or kept on as part of your protein product. So when you have a gene that is activated in your cell, so the cell needs to make this particular protein, the first step is making the messenger RNA. This is complementary to the gene, so just like your base pairing, there's a T on the DNA, then it's going to be an A on your RNA. If there is an A on your DNA, it's going to be U. So this one's where that pairing is a little bit different because of the RNA. So you're going to go through that whole sequence for just that section of DNA to make your mRNA. So your mRNA is going to move from the nucleus into the cytoplasm where you're going to make your proteins. So your process of protein synthesis is going from your DNA to make your mRNA copy of just that gene, so not the whole DNA, just the gene that you're going to make your protein from. And then from this mRNA, you're going to make your protein, so coding for the different amino acids to make up that protein. So the process goes from DNA to mRNA, which is called transcription, and then that occurs within the nucleus. The next step, mRNA to protein, that's called translation. That occurs into the cytoplasm. So another way of thinking this is like, let's say you're sick and you go to the doctor. So your doctor has all the information possible that you need in order to diagnose your condition. So like your DNA, having all of your blueprints for making your protein in your body. So the doctor is going to give you a prescription or to transcribe something for a particular medicine. Then you take that prescription and you'll take it to your pharmacist, and your pharmacist is going to translate whatever your doctor put on that paper into your whatever medicine that you're going to take for your condition. So now we're going to go through the steps for going through transcription as well as translation. So for transcription, you're going from DNA to mRNA. So in order to do this, there's an enzyme involved called RNA polymerase. That enzyme will bind to DNA, help open it up, and start reading the DNA to create your new strand of RNA, so just the copy on that particular gene. The start signal for a particular gene is this repetitive, what's called a ta-ta sequence. So it's a repetitive TA for a bunch of different nucleotide coding. Not sure where your RNA polymerase knows to bind. So after it binds to that, then it'll start reading the bases and starting to build the mRNA from that. So wherever it finds a C on the DNA, it'll add a G. Where it finds a G, it'll add a C. And where it's a T, it'll add an A. Again, the difference between when you're making base pairing for DNA versus RNA, when there's an A, it'll put a U instead of a T. So here's a little green RNA polymerase going through and making the pairing. So here's the one part where it is different. So the next one that be for this slot here will also be a uracil. So as the RNA polymerase enzyme is moving through DNA, it opens it up, and then it also rewinds the DNA behind it. You can transcribe the gene multiple times, so making multiple copies of your RNA if you need to make a bunch of a particular protein. But still, once your RNA polymerase goes through that gene, then once it hits that stop signal, then it will stop, detach from your DNA, and then if you need to make more, it can reattach to that gene and continue to make another copy. So when you have your mRNA sequence, which is just your gene, of course, again, your gene can code for multiple proteins. So you have what's called your pre-mRNA, which is an immature mRNA that is produced. And some of the pieces of this gene, so what I mentioned earlier, the non-coding portion of your DNA, those introns are removed before that mRNA can be translated into a protein. The exons stay, and that part is what's translated into a protein. So most likely these introns are used for regulating the protein synthesis or protein production. So the removing of the introns occur inside the nucleus to cut the RNA open, remove those introns, and put it back together. So one way that your mRNA can be reorganized to make different protein products is by, as you're removing, so the darker sections are our introns that are going to be removed, and then you can reorder your exons in whichever way you need to make different protein products. So you could have a protein that's A, B, A through F for the exons, or you can have proteins that are only part of these different letters, lettered exons, and then reorganized if you need to. So it depends. That's one way of how your gene can code for more than one protein. Okay, next step. So translating what was transcribed. So taking our mRNA copy of our gene and making it into a protein. So converting the language of nucleotides into the language of amino acids. So there's three main participants in translation. So again, we have our mRNA, which is our copy of our gene, moves out of the nucleus and into the cytoplasm. There's a site, there's a protein cap on it that is a recognition site for our ribosome. So here in the purple is our rRNA, which is our ribosomal RNA, and that's where the protein production is going to occur. Here in the little squiggly lines, those are our tRNA that's going to bring the amino acid to this site and continue helping to build the protein chain. So once it gets rid of its amino acids, then it gets released out. So on your transfer RNA, in order to pair the correct amino acid to the codon pairs, it has what's called the anticodon. So it's the base pairing to the codon on the mRNA. So when these link up, then your ribosome's able to move over. These two amino acids going to join to this new amino acids. And as it shifts, this tRNA is going to move to our little exit site and go off to bind to another amino acid. So we start off with our mRNA that has a leader sequence. It's again site for ribosomes to bind to it. And as the ribosome is going through your mRNA sequence, instead of the ribosome moving, it actually pulls the mRNA into it. So, for example, the ribosomes that are docked to our rough endoplasmic reticulum, they of course don't move. So in order to read the mRNA, it pulls it through like a rope. So once it keeps pulling, eventually it's going to start at your AUG start codon, and that's where it starts creating your protein. Again, methionine is the first one. So here's a blow up of our tRNA, which is again is our transfer RNA. So this end will bind to the amino acid. Here's our anticodon at the bottom. Even though this has some kind of a little bit of pairing to create this tRNA shape, it's still one strand of genetic material to make that tRNA. So as you're making your proteins, there's 3 steps. Your initiation phase, so adding your ribosomal RNA to your mRNA molecule. Again, starting with your leader sequence, and then you have your binding to it once you get to your start codon. Okay, so we have our leader sequence. The small ribosomal subunit will bind to the mRNA there, and then it'll keep pulling it through once it gets to the start codon. The large subunit, so the top portion of that rRNA will bind to the mRNA in pink, and then you start adding, reading those codon bases and pulling that mRNA through to continue adding more amino acids until you reach that stop codon. So next step is elongation. So as your rRNA, so your ribosomes pull your mRNA through its complex, you'll have tRNA, your tRNA will bring additional amino acids to the site. Then a peptide bond is formed with the next amino acid. Then it pulls it through. This tRNA that's in this site moves to our exit site after it releases those amino acids. So here's our growing peptide chain here. Once that peptide bond is made, everything shifts. So now this tRNA is going to be in this location, waiting for another new amino acid to be delivered so you can make another peptide bond. So you're going to continue this until you get to that stop codon that does not code for an amino acid, and that's how your ribosomes know where to stop making the protein. So once you've reached that stop codon, the ribosome will release and the finished protein will break away from that site. So for translation, you can actually make a ton of proteins at the same time. So here we have a bunch of different ribosomes making protein chains off that single strand of mRNA. So some of your proteins, instead of just being made freely in the cytosol, they're actually made on the surface of your rough endoplasmic reticulum, and your amino acid, your peptide chain is actually fed into your endoplasmic reticulum so that it can be packaged into a vesicle. So these are your vesicle packaged proteins that will eventually be modified later in the Golgi complex. So that will be your start of your next YouTube, or at least next video.